A major component of modern ecological research focuses on understanding what influences the abundance of organisms within different populations and why that relative abundance, or abundance in general, changes over time. Now, kind of going back to our definition of a population is a group of individuals of the same species that occupy a specific area over a certain period of time. Now, when we look at populations in nature, so how they exist in reality, they exist in a continuous state of motion, per se. In other words, they're essentially in this continuous flux. Now, the patterns that arise in distribution and abundance, they arise from interactions between various types of factors, or these dynamic processes that operate to add or subtract individuals from populations. These same dynamic processes can also hold certain populations within confines or in limitations of other populations. If you think about predator-prey interactions. So essentially, all populations within an ecosystem operate and interact with one another to maintain this equilibrium, per se, where you go through changes in relative population sizes based on the population size of surrounding species, or the presence of different species. You'll also have these kind of ebb and flows as far as population sizes in response to changes in the environment. Right? So it's a very dynamic living system as far as the interactions or the sizes, the distributions of different populations. Now, this whole area of ecological study is what we call population dynamics. The actual term population dynamics refers to how populations of specific species change over time. So the study of a species' population dynamics is primarily concerned with the factors that influence either the expansion, declination, or maintenance of populations. So in general, we refer to populations as being dynamic. Again, there's these dynamic processes that are held in operation that contribute to the changes in size and composition of populations over time. Now, there are several factors that allow for these fluctuations. Again, it can be environmental changes. It could be the presence of new species that may be deleterious to the survival of another population. The kind of major or the majority of your population fluctuations are the result of the net difference between additions to and subtractions from the population. Now, there's three major factors that can kind of manipulate this and affect population size primarily, as well as composition. When we talk about population size, what I'm referring to is the number of individuals that are present in a population. So anyway, these three major factors are birth rate or natality, death rate or mortality, and movement. Movement or migration deals with immigration and emigration rates. Now, these deal or involve either the addition or subtraction of individuals to or from a population. Your additions are from natality, which again is birth rate, and immigration. Both of these add individuals to the populations. So your populations will grow due to both of those factors. On the other hand, factors that serve as subtractions from populations, those will be mortality or death rate and emigration. Both of these, again, remove individuals from a population. So you see declines in population size and number or density due to deaths and immigration. Immigration just occurs when individuals will leave a population to join another. Now, ignoring immigration and emigration, growth itself is primarily affected by your birth and death rates. The actual value of the effect of these two factors depends primarily on how many individuals are being added or removed to that population. In general, you can kind of measure the effect of these factors by calculating what's called an intrinsic rate of increase, which is also denoted as R, a lowercase r. This is simply the difference between birth rate and death rate. So it's equal to B for birth rate minus D for death rate. It's also known as the innate capacity to increase. Now we'll move through some other specific dynamic aspects of different populations. As I said before, the major factors influencing your populations as far as size and composition are those three factors of birth rates, death rates, and emigration or immigration movement per se. But kind of going more in specific with some other factors as well, you can really look at dynamic aspects across time and space. Right? So you can group them accordingly. There's spatial dynamics, which consider such things as the influence of dispersal on distribution and abundance. You also have temporal dynamics, right? So that's that time component, which focuses on more of the patterns of survival and reproduction in populations and how that affects distribution and abundance. So we're going to cover each one of those and move through each, starting with the spatial dynamics, focusing solely on aspects of dispersal. Now, a lot of information can be gleaned or obtained by looking at and evaluating the distribution of individuals and observing how distribution influences or affects abundance and density. Density is the number of individuals per unit area. So how does that measurement change based on relative patterns of species and population distributions? Species dispersion patterns, or their distribution patterns, they show spatial relationships between members of a population within a habitat at some particular point in time. So in other words, they show whether members of a species live close together, maybe it shows that they don't live close together, rather they live farther apart, but it can also show what patterns may be evident when these individuals are spaced apart, as kind of gleaning behavioral patterns that may arise, or what behavioral patterns cause or serve as causation for those distribution patterns. Now, there are kind of three primary types of arrangements as far as dispersion that are observed in nature. Individuals can be arranged in populations either randomly, right? So this is when individuals are kind of dispersed randomly across the habitat with no predictable pattern. Individuals could be clumped, which is when you have groupings of individuals here and there around the habitat, or you can have uniform patterns, which is where the individuals in a population are more or less equally spaced apart from one another. So we're going to take a peek at each one kind of more in depth. So starting with your random dispersion patterns. This is what you can kind of think of as taking a handful of seeds and bird seeds and just randomly throwing it to a flock of pigeons. Now, each one of those seeds will land randomly across the ground, and they fall at no predictable pattern. This one isn't going to fall this close to this one for this reason, etc. You can't predict where each one is going to land because there is no kind of formula that's guiding such action. In this type of dispersion, the position of each individual is independent of the others. Again, this individual isn't located or inhabiting this specific point because of some external factors that are placed by the position of its neighbor. Again, it's all random placement as long as that environment is favorable. So you see this a lot with plants, particularly plants that are wind dispersed. So think of dandelions or the seed pods of maple trees. They all utilize wind dispersion to carry their seeds to other locations in a habitat or different habitat sites to establish new populations. And those seeds will germinate wherever they happen to fall if they fall in a favorable environment. Now they're, again, carried away randomly. They're moved randomly to a habitat. It's not necessarily that they are brought to a favorable habitat. Sometimes they can be brought to one that is unfavorable, and therefore they would not germinate and establish a population. But that's what is meant by random dispersion. You could also see this when you have kind of this random speckling, per se, of resources across a habitat. Now, clump dispersion, this is where you have kind of pockets or little groups of individuals clustered throughout favorable habitats. So it's almost like you have small subcolonies, per se, of a whole population. Now, this will occur when the quality of the habitat is patchy or you're working with organisms that are social. So kind of talking about some examples with plants. So with plants, you don't quite so often see this with plants. It's more with social organisms. But with plants, an example would be oak trees. These trees just drop their seeds straight to the ground, so you kind of get these kind of relative stands of oak species that form these clusters across a habitat. This can also form if there's a barrier to dispersal. So something's preventing those seeds from being carried to other areas of the habitat or different sites within an ecosystem. So they're forced to stay in those groups. Now, as I said before, you largely see this with your animals, your social organisms. These would be schools of fish, herds of elephants, etc. And there's several advantages to living in groups. Now, not everybody does it because there are some disadvantages, but in general, for some species, there's advantages that arise as far as mating, protection, right? So if you think of a pack or a colony of meerkats, they work together to watch for predators and patrol territories. And by doing so, it actually increases the inclusive fitness of that population. But again, this isn't for everybody, but you see it largely with these groups that are social and or mechanized by aspects of kin selection. So they are working to protect their offspring and promote the growth and flourishment and the following reproduction of their offspring. You'll also see this kind of as a byproduct or a function of what's known as habitat heterogeneity, which is basically where you have a mosaic of different habitat types across an ecosystem or a landscape area, or patchy resources, right? So this type of dispersion pattern provides a lot of information, not only about kind of simple density measurements as far as, all right, where's there going to be low density species or high density of species, but rather how these populations and individuals interact with one another. And you can kind of use that information to determine when you put these same species or individuals of these clumped species in solitary kind of populations, or in more about this random dispersion, how that might affect their mating success or hunting success. Again, for social species, that may come far more difficult or become more difficult if they're put into a random dispersion than if they were solitary species. The last one is uniform dispersion. Uniform dispersion is a little weird because you don't see it quite so often, noticeably per se, in nature. It does exist, but it's hard to find. You have to look for it. Uniform dispersion, think of lines of corn or rows of corn or wheat in a farmer's field. You have these almost kind of linear lines where each individual is, again, more or less equally spaced from one another. Now, how the heck does this happen, or why would that happen in nature? Again, it's hard to find, but it exists. Usually this will arise when there are negative interactions occurring among individuals of that species, and those negative interactions cause them to space out evenly, almost to avoid one another. So for example, if there was competition for an evenly distributed resource like water, or if there was a high element of territoriality that was operating in a population, right? You see this with penguins. So penguins will follow or display uniform dispersion patterns because each individual maintains a defined territory that they will only share with their reproductive mate at that time. Otherwise, each nest is essentially some uh, sort of distance measurement from all surrounding nests, from all of its neighbors. So those very specific territories around each nest site, causes these very even placements of nests. So you get this uniform dispersion throughout a flock of penguins. More commonly, you see this with some really cool plant species. There are some plants that um, allow or are examples of a really cool phenomenon known as alleliopathy. This is where I like to say plants use chemical warfare. They actually secrete different substances and toxic chemicals that can inhibit the growth of nearby individuals. And what they end up creating is almost this ring of zero growth or limited growth around the base of that plant. Ranges that kind of zone of inhibition, you can think of it. Ranges in size based on the plant species. But you see this with sage and also with black walnut. If you go into a field and you find a black walnut tree, they're easy to find. Look at their bark characteristics and look for that giant turd-looking thing on the ground. Next time you find one, look at the base or look around the base of the tree within kind of maybe not 5 foot, like a 3 foot radius around the base of that tree, the trunk of that tree. You will find almost no growth around it, except for some grass, maybe some weeds, some kind of hardy plant species, but no real defined understory besides those small grass-like species. The reason for such is because their roots secrete these toxic chemicals into the soil that prevent other plants from being able to germinate, or seeds that land there from being able to germinate at that site. And by doing that, the black walnut tree doesn't have to compete with other plant species for water, for soil nutrients, for sunlight, for space, etc. So when you actually have a large field full of black walnuts trees, you'll notice that they are somewhat evenly spaced from one another, because they will also prevent one another from occupying spaces too close to each other because of those chemicals. So it not only inhibits other tree species, but it will inhibit other black walnut individuals from cropping up too close to others. Really neat stuff. Now, kind of thinking about the different causes that create these distribution patterns across habitats, or why certain patterns will change in time, or you'll see certain populations of species following one sort of distribution pattern in this habitat type and a different distribution pattern in a very different habitat type, if you're talking about like a generalist species. What could be some of those causes mechanizing those differences? Well, there's a variety of causes. There's proximate causes and ultimate causes. Proximate causes refer to events that are kind of immediately responsible for causation, right? So They are kind of the closest responsible factor or immediate reasoning for some observed event or result. You can think of them as almost being like that momentary, superficial cause. A lot of these are physiological and or behavioral mechanisms, as well as climatic. So, for example, um, if you looked at a local population, right, and there was a high degree of growth during a very resource-rich period of time, that could cause crowding. It could also lead to kind of strained food availability. Those would both mechanize changes in distribution. Right? In that case, a lot of individuals would probably move out of the original population to seek confines elsewhere where they can establish a new population in a more favorable habitat. You also see this if kind of thinking about your predator guilds in Africa. Talking about your major carnivores, so lions, hyenas, leopards, cheetahs, African wild dogs. They all show these really interesting changes as far as distribution based on the presence of other predators. So cheetahs, for instance, actually modified their hunting times to very, very close to midday period, which is unhealthy because it's the hottest part of the day, but they do that to avoid competition with lions and hyenas. So that's a change in behavior. That's an approximate cause, but it's caused them to have these different distribution patterns when they are in the presence of these higher predator or carnivore species. Your ultimate causes. So these are a stark contrast to proximate causes in that they're more of these higher level, somewhat distal types of causation. Usually they're thought of as being the real reason why something occurred. A lot of them have to do with evolutionary adaptations, either to increase fitness in an area, or more specifically, to prevent inbreeding and mitigate inbreeding depression from occurring. By doing that, by dispersing in ways that minimize inbreeding, you can increase the fitness of a population by increasing genetic diversity. So those are just kind of differentiating between the different mechanisms that serve to basically define the types of distribution patterns that populations will exhibit in different areas. All right, so that really wraps up the spatial dynamic processes as far as population density or size and composition. So moving now from those areas of spatial processes into temporal dynamic processes, this really looks at how patterns of survival and reproduction influence distribution and abundance. At kind of any point in, or any particular point in time, a population can be described by its size and density. Right? That's how we, kind of what we just did as far as talking about changes in density. or factors that may cause that. Now, to actually study the true dynamics of a population, scientists have to employ a field known as demography. Demography is the statistical study of factors that determine the size and structure of populations through time. So, in other words, it studies how populations change across time. Now, what are these factors? Well, they're the same factors that really are the primary influencers as far as population size and composition. It's birth rate, death rate, and instead of immigration, you have life expectancy. Right? So, how many individuals are dying? How many individuals are being born? And what's the relative survivability of each individual? Now, each of these measures individually, particularly birth rates, they can be affected by other population characteristics. Right? They can be affected by the density of a population, or if there's a sudden boom in population size in a confined area, that may cause overcrowding. If you have these larger sizes, right? So if you use size as a factor, you may have a higher amount of birth rate because there likely, or there may be more reproductive individuals present. In the same notion, kind of on contrast, a large population size can also result in a higher death rate because there's a higher risk or a larger degree of competition, disease, and waste accumulation. There can also be kind of strained or influenced, more I should say, impacted reproductive encounters between individuals, which can either increase or decrease birth rate. So those specific measures themselves, again, can be affected by other population characteristics as far as size, composition, and relative other dynamics. Talking more about spatial dynamics. But by knowing different aspects of demographics, scientists are able to make predictions about the future of a population, which is very, very important. Now, if you are to make these predictions, you need to know some specific information. Right? You need to know age structure. Right? So the number of living individuals of each age across that population. You need to know the number of individuals of different ages that are likely to survive into the following year. And you also need to know the number of offspring that may be yielded or produced by each individual or each female of that population, and how many may be produced at each age, right? So how many offspring may be produced in a female that is 15 years of age versus one that's 30 years of age? And what is the likelihood of survival the following year at those two ages? How do they differ? You also need to know how many individuals of different ages immigrate and emigrate each generation. When we talk about a generation is the average time between a mother's first offspring and then that of her daughter. So how much movement is occurring between that time? When we talk about kind of making these predictions and the information you need to know, these are all honing in on factors of what's known as population growth rate. Right? So what factors are influencing the rate at which a population can grow over an interval of time. And those factors are most of the information that I just talked about, but also sex ratio, right? So what's the relative number of females versus males in a population? Going back to that aspect of generation time, right? So this goes back to, all right, so what's the number of offspring a female can produce at a certain age, and how much time exists in between each generation, right? So how long is it before you have the second generation or the third generation of offspring occur? And also, what are the reproductive ages? So at what age do your females first become reproductive, and therefore can you have the first appearance of offspring generations? They're all important factors that will influence the rate at which your population can grow in size, as well as the rate at which you can have changes in population structure. Now, to predict if a population, hello, Figaro. To predict if a population will grow or shrink in size, ecologists need to know what the birth and death rates are for different organisms at different ages. And they also need to know the current age structure or age makeup of a population, as well as the sex makeup. So going back to what I had mentioned before, as far as knowing the sex ratio, age structure, how many fertile or reproductively viable females are present at that time in the population. And ecologists will use different tools to describe or evaluate the present state of a population, and also make predictions about how it may grow in the future. The kind of primary tools that are used that I will be discussing are life tables, survivorship curves, and age distributions, or age structures. Life tables summarize birth and death rates for organisms at different stages of their lives. So it looks at patterns of survival. Survivorship curves, kind of honing in on their name, also look at patterns of survival. But these are more of visual representations of life tables. They're graphs that show what fraction of a population survives from one age to the next. And then the last type that I'll be talking about are your age structures or age distributions. These are essentially snapshots of a population in time, showing how its members are distributed among age and gender or sex categories. So we'll start with your age distributions or age structures. Again, these are used as one of the tools to help understand population structure and also make predictions about how it may grow or change in the future. The age structures are also known or called population pyramids or age pyramids, though they are not always shaped as pyramids, so don't be misled by that. But the diagram in general operate to serve as these kind of visualize or explain the age structure of a population by illustrating the distribution of different age groups within the population across gender. So these are looking at several different images of different age structures. Usually you'll see this kind of either pyramid or conical shaped structure that's distributed across the x-axis, where the x-axis is representing population size or percent population in millions. You always have kind of this separation in half of the structure where the left hand side represents males and right hand side represents females. And either along this notable y-axis or sometimes kind of right in the middle of the two gender clusters, you'll see an axis that highlights the age brackets or the age categories of individuals in that population. Right? So you may have 0 to 1, 1 to 3, 3 to 5, etc., etc. Now, kind of interpreting this, right? Okay, so you're looking at this shape. What are you really looking at? So, as I said before, left side are males, right side represent females in the population. Usually you'll have, or if you see a wider base, I should say, that indicates the birth rate. As you move up in the ages, you will see kind of variations in the lengths of these bars. Each of these bars, the colored part, indicates the percentage of the population or the number of individuals in the population of that age that are working or surviving. And from there, kind of looking at, okay, so here's your starting population number, looking at the indentation of each bar highlights or insinuates or indicates mortality, right? So the number of individuals that have passed on or have died either of natural causes or because of disease, famine, etc. So you can essentially more or less get an indication of life expectancy or survival probability for each age, as well as in general what the relative life expectancy is for each of these genders in this specific population. Now, there are 3 levels per se in these structures. There are kind of 3 age or reproductive levels that are shown. Usually the population is divided into pre-reproductive, which is your lowest stage, reproductive, and then post-reproductive stages or phases. In most of your populations, you can determine or it will highlight at what age the females will become reproductively viable. So if you have more individuals that are of this reproductive stage, there's essentially you have increased number of potentially reproductive individuals that are present, which could increase birth rate. So you can start making inferences about population trends that may arise from these relative age structures. And as I was saying before, the shape isn't always pyramidal. It can vary, and the shape of the diagram can show you if a population, or in this case a country, whether it's rapidly growing or if it's growing slowly or maybe not growing at all. There are some that may show 0 growth or stable growth, in which there's no growth no change, no increase or decrease. So here on the far left side, this structure, so if you see this pretty, it looks almost like an equilateral triangle shape, that's representative of rapid growth. You see this a lot when we're talking about human populations. You see this a lot in uh, um, the least developed nations, because they'll have uh, relatively high birth rates, but very low survivability at higher or older ages. This isolateral triangle shape, this structure is representative of slow growth. So you see this with the transitional nations. Right? These are stable populations, meaning there's no growth or decline. So they're both in equilibrium. Right? Some examples would be Sweden. Here it also says Australia, Canada, and the United States. This more of a conical shape, where it's more rounded rather than pyramidal. This represents zero growth or declining growth, which you see in most of the developed nations, or I should say, most developed nations. So these are nations that are kind of moved out of that transitional stage, and actually have started to decrease birth rate. So examples would be China, Singapore, Italy, Denmark, Austria. It's almost it's not so much that they're held in equilibrium anymore, but it's actually going in the opposite, where the mortality rate is starting to surpass birth rate. So if birth and death rates are equal, right, your population will follow this. Your population remains stable. However, if the population size increases such that birth rates increase or birth rates exceed death rates, then you will have an increase in population size. So it would be more representative of this rapid growth that you see in a lot of the developing nations. Population will decrease, right? On the other hand, if birth rates are less than death rates, which is what I was saying for this zero growth. So zero growth or negative growth occurs because your birth rates are less than death rates. There's a higher proportion or ratio of mortality versus birth. So not enough individuals are being born to replace those individuals that are being lost from a population. Now, this obviously your birth rates and death rates are very important or influential in determining those kind of growth trends. But in the same instance, life expectancy is also important. And again, age structure is important. Life expectancy obviously is the length of time an individual remains in the population. Now that impacts local resources, reproduction, and the overall health of a population. So usually as it's great to have a greater life expectancy, but that does cast certain detriments or influences to that population. Also, in general, when you look at age structure, most of the reproductive potential lies with young individuals, right? If you look back at the division of your population into pre-reproductive, reproductive, and post-reproductive stages, this area here, the reproductive phase, this is where that highest potential for population growth rests, right? Because these are all individuals that have the capacity to produce offspring, which can then replace individuals that are lost. Now, that birth rate may be greater than mortality rate, it may be lesser than mortality rate, but in general, you can look at that age structure and determine or predict what trend the population may follow in the future based on the ratio of young individuals to older individuals, right? So if a population consists mostly of young individuals, most of those individuals have a high survival and reproductive rate, then assumingly, or you would predict that that population would likely increase over time, because again, those individuals they make up the majority of the population, and since they have a high survival rate, they will continue to live a certain number of years, or they have a higher probability of living a longer number of years following that moment in time. And they also have a higher probability of reproducing in the future, as well as reproducing maybe multiple times. If you, on the other hand, looked at a population that was comprised mostly of older individuals, a lot of times older individuals have lower survival and reproductive rates, it's likely that that population will decline over time because you do not have a sufficient foundation or stand of reproductive potential to either maintain the stability of that population, so a stable number of individuals to bring in offspring that will replace individuals that are lost in time, or there's not enough individuals in general to create a high enough birth rate that brings in offspring. Again, that's what you're seeing, or what's being represented by negative or zero growth. So by looking at those ratios with age structure, you can make predictions about what trends may occur in a population over time. And that's extremely important when you're looking at different wildlife populations, particularly populations that are threatened or facing endangerment, seeing kind of what the birth rate and survivability of offspring are in those populations allow you to infer, okay, is this population going to have the potential to restore those numbers over time, or will that population face further declinations because it cannot reproduce fast enough, or there aren't enough individuals to reproduce and create offspring that can survive in the population. So those are your age structures. Those, again, is one tool that ecologists use. Another tool that I mentioned are life tables. Life tables provide important information about the life history of an organism and summarizes the probability that an individual will survive and reproduce at any given time interval over the course of its lifetime. And they're actually modeled after actual tables that in modern times, so today, stamps are used by insurance industries and companies for estimating human life expectancy. Life tables in general have been around for thousands of years and have been used for thousands of years. Historically, they were used by the Romans to predict food needs. As I said before, today they're used by your insurance companies to predict lifespans. Now, when you look at life tables, they divide the population into age groups, and some of them will further divide that population not only into age groups but also groupings of gender. And they will show again how long a member of that group is likely to live. Right, so an individual of this age group and of this gender is likely to survive or live for this many of consecutive years following, and has a probability of X amount for surviving. So it's essentially charting or predicting the probability of surviving versus dying, and how likely an individual of a certain age is to survive onto the next year. Now, it sounds very morbid, and it kind of is, but it yields valuable insight as to the potentiality of a population based on age distributions, mortality rates, and also sex ratios. Right, so many of these tables will include the probability of an individual dying before their next birthday. Right, that's mortality rate. Very morbid, but it's useful information because, again, sometimes you run into trouble if you increase life expectancy as far as being able to allocate enough resources to provide for that increased number of individuals that are now living for a longer period of time. Some tables will also show or often show the percentage of surviving individuals dying at a particular age interval, and the life expectancy at each age interval. Now, in general, kind of going back to how this fits in with ecology and kind of conservation biology, the average number of offspring left by a female at each age, together with the proportion of individuals surviving at each age, that can be used to evaluate the rate at which the size of the population changes over time, or the predicted rate at which you can expect change. And then these rates can be used to estimate and predict population growth or population decline in the future, and evaluate the effects of different conservation efforts. Right? So if you're talking about an endangered species here, and we're using Vortex, which is a certain software that allows you to model, kind of throw in different factors as far as birth rate, death rate, life expectancy, number of fertile females, number of old individuals, age groups, etc. We can throw all these factors into this Vortex program, and it will tell you or generate predictions of population growth over a certain number of years, so you can see whether it's growing or predicted to grow or decline. With endangered species, that's important because then by using that information, you can say or evaluate whether these conservation efforts are actually fruitful, if they're actually making a difference, and a positive difference, or if things need to be changed. It also allows us to start honing in on species that may need help longer down the road. So here, this is an example of a life table. This is a life table that was created from a study on Dall mountain sheep, which is a species of sheep that are native to northwestern North America. Now, the population is divided, it's not divided into gender, but it's divided into age intervals, represented by number of years. So some will show number of years, some may be number of days, some may be hours if you're talking about very short-lived species. For these guys, these are K-selected, so they have very long lifespans or longer lifespans. So their age groups are, or age intervals are numbers of years. Now this column here, this is showing mortality rate per 1,000 individuals. Right? So this population, kind of at the start, or the original number of offspring at this start of this generation was 1,000, right? So the mortality rate per these 1,000 individuals is highlighted in this column for each age interval, right? And it's based on the number of, you can use this formula here. So mortality rate is based on the number of individuals dying during that specific age interval, right? So that would be this column here, right? So in, we'll say this 1 to 2 year age interval, right, of those original 1,000 individuals that were born, 12 are predicted to die, or in this life table, 12 die. So you can take this number divided by the number of individuals that are surviving. Right? Here's your number of surviving. So of those 1,000, 801 are continuing to survive, so will survive into the next year. And then you can multiply that by 1,000 to get, again, mortality rate per original number of starting offspring. So, for example, if you look at the age interval for 3 to 4 years of age, 12 individuals of the 1,000 die out. Actually, sorry, 12 individuals of the 776 individuals die out, because that's how many have survived to this age bracket. Of those, 12 will die. So in general, there's 776 individuals that have survived from the original 1,000. Of these 776 surviving, 12 will die at ages 3 to 4, or when they are of that age. Now that number, when you divide 12 or divide 776 by 12, you multiply that by 1,000 to get the mortality rate per 1,000 individuals. So in this case, that mortality rate, 12 divided by 770, or 776 divided by 12 times 1,000 is 15.5. So that's how these tables are generated. And again, you can use this information to predict the number of individuals in a generation that are predicted to survive at each age, whether there's a higher percentage of younger individuals or older individuals, which one has a greater survivability, and the life expectancies. Right? So these are the general elements that you find, and kind of their symbols. These will crop up later, and I'll explain them again. So the last tool that's used are survivorship curves. They're essentially visual representations of your life tables. these tools are graphs, or they graph the logarithm of the number of individuals surviving at each age interval against a measure of time or age. Again, usually these graphs are compiled from data derived from your life tables. And they're a very powerful visual tool for understanding the patterns of survivorship and mortality in populations. Now, when we talk about survivorship, okay, what does that word mean? Well, survivorship refers to the proportion of individuals produced that survive on average to a particular age. So in general, your survivorship curves, based on that definition and the curve nature themselves, they're used to show the distribution of individuals in a population according to age. Now, there are 3 types of curves: type 1, type 2, type 3. Not very creative, but very easy to remember. So type 1 curve, in this curve type, right, so this is the curve that's in blue, survivorship for the majority of life is relatively high. Specifically, a high percentage of offspring survive during their early and mid years. Right? So this is life expectancy based on numbers of years. This is the log number of individuals that are surviving. So during these ages, you have high survivorship. In general, these ages are of the early to mid years. Death, right, which is distinguished by this low survivorship, occurs predominantly in older individuals. But in general, most of your individuals that are kind of representative of these type 1 survivorship curves will approach the maximum lifespan of a species, meaning most of them will reach that predicted maximum lifespan, whether it's 100 years or 89 years, most of them will reach that limit. Now, the species that you often see exhibiting this type of survivorship, they usually have small numbers of offspring at any one time, and they invest a high amount of parental care into those offspring to ensure that they survive. So that kind of helps to explain why there's such high survivorship of offspring. There's such a high degree of parental care that's invested, but it's invested for good reason. A lot of these clutch sizes or nest sizes of these species, again, are small numbers of offspring. So they invest a lot of care into these small offspring yields in order to increase the survivability or probability of survival. So some species that are representative of this: humans, primates, most of your mammals in general have a type 1 survivorship curve, largely because death primarily occurs in the older years. There's very high survivorship and survivability in the younger to mid years. Now, type 2 survivorship curves, these represent or occur where individuals experience relatively constant or stable levels of survivorship over their lifetime. And what I mean by that is essentially the probability of death at any age is equal, right? So this individual is equally likely to die at age 1 as it is to die at age 15. Um, there's no real skew as far as survivorship, um, more towards the younger or older years. It's steady throughout that lifetime. So birds, especially your songbirds, are examples of this type 2 survivorship curve or intermediate curve, um, because they die more or less equally at each age interval. Um, when you talk or think about this, the type of species that follow or are examples of these types of survivorship curves, a lot of these are organisms that have relatively few offspring, but also do provide significant parental care. So very similar parenting behavior to that of the species that you find the type 1. Now type 3 curves, right, the last type, these result from high death rates that occur very early in life, with higher rates of survivorship occurring after maturity, right? So it's essentially an inverse of type 1. There's low survivorship in early years. Once these individuals kind of get past this horrible point in life and reach maturity, their probability of survival increases. So there's actually high survivorship as age increases to a certain degree. Trees, most of your plants species have this type of survivorship, because very few survive the younger years, very fragile. But after a certain age, the individuals become much more hardy and are much more likely to survive. And when they do survive, they're more likely to survive for a relatively longer period of time. Marine invertebrates also exhibit this, as well as most fishes, or fish species. Most of the organisms in this category typically have a large yield of offspring, or high offspring production rate per se. But once those young are born, very little parental care is provided. So the offspring are essentially on their own and vulnerable to predation, which is why they have low survivability. There's a high mortality because of the increased predation due to decreased parental care. But their sheer number at which offspring are generated per generation assures that enough individuals will survive in order to perpetuate the species. So these are a lot of your either species that have very high reproductive rates, very short gestation periods, or are selected species. So think of a lot of your insects. So this is looking at the survivorship curve that was generated using the data compiled in the life table for Doll sheep. And based on this curve, you can see that they exhibit a type 1 survivorship curve where you have high survivability or survivorship during the early to mid ages, with lower survivability as age increases. So as these individuals get older, their survivability or likelihood of survival decreases. Alrighty, so when we talk about kind of monitoring the rate at which a population can change, right? You look at a population and you want to actually determine, one, is this population going to grow? And two, what is the rate at which that change will occur? That's going into this area, the rate of population change. You're not so much looking at survival anymore. Survival, or estimating survival, comes into play for determining whether, or kind of the movement of population change. Are you going to have population growth or population decline? What's going to happen or what's predicted to happen in the future? All of that uses estimates of survivability and mortality. When we talk about kind of honing in at the rate at which change will occur, we're again not using survivability so much, but rather birth rate. Right? So this deals with tracking birth rates in a population to evaluate density and how density may change over time and at what rate that change will occur. And it's very similar to the methods that are employed to tracking or for tracking survival rates. Now, when we talk about birth rate, right, what is birth rate? Birth rate represents or is equal to the number of young born per female in a period of time. Now, when we're talking about sexually reproducing populations, there's important information you need to know, right? You need to know the average number of births per female of each age class, as well as the total number of female individuals in each age class for that population. And in general, what ecologists will do is they will combine information from life tables with something known as a fecundity schedule. Fecundity schedule is a tabulation of birth rates for females of different ages in a population. And it'll essentially, you can think of it as representing fertility rate. If we combine the information in fecundity schedule, try to say that 10 times fast, with that in a life table, we can estimate several important characteristics of a population. These characteristics being net reproductive rate, or R0, geometric rate of increase, represented by lambda, generation time, and intrinsic rate of increase, also known as the per capita rate of increase. We talked about this guy previously. These can all be used to yield important information as far as estimating and understanding whether the population is growing or declining and at what rate that change is occurring. Now, we'll talk about each one of these estimates or parameters specifically, but we'll talk about them more as we approach them. So to start, the major thing or major facet of information that is probably of the utmost importance is understanding net reproductive rate. Net reproductive rate is or represents the expected average number of female offspring which a newly born female will produce during her lifespan. Right, so you have this, say we're working with a population of sheep again, and this little baby girl was just born. Actually, we'll work with horses since we just worked with sheep. Gotta share the love. So you have a little filly that is born. The predicted or expected number of female offspring that she is expected to yield during her lifetime, that is net reproductive rate. And this measure is an indication of whether a female will replace herself in a population. Kind of what the likelihood of that is. So once she passes, what's the likelihood that there will be an offspring that she has yielded that will kind of serve to replace her in the population. Now, if all females survived to the oldest possible age for every population, the net reproductive rate would simply be the sum of the average number of offspring produced by females at each age. However, that's not what occurs in reality, right? In real populations, some females die at every age. Some will have higher death rates at certain ages, but generally at every age, a certain number of females are dying, or there's mortality at every age. So you have to generate these net reproductive rates based on those differences across ages or age classes. The net reproductive rate for a set cohort. A cohort is represents a sample. So it's a sample of the true population that you're working in. But that the rate for that cohort is obtained by multiplying the proportion of females surviving at each age. Right? That's represented by LX, by the average number of offspring produced at each age, which is represented by MX. And then adding the products from all of the age groups together. Right? So the sum of all of these, the products of all of these age groups, that yields or allows you to calculate your net reproductive rate. Now, when you get the value for net reproductive rate, right? So you get a value of 0.2. Okay, what does that mean? Well, you compare the values you obtain to 1. If your R value, so if your reproductive rate is less than 1, the population will decline. A net reproductive rate that's equal to 1 or 1.0 indicates that a population will remain constant. It's neither increasing nor decreasing, but rather the numbers are being replaced exactly, and that population is therefore stable. If your R value is greater than 1.0, that indicates the population will increase. There's actually more offspring being produced than what's needed to replace that female when she's lost. So that's net reproductive rate, how you calculate this indice, and how you use it to determine the trend of the population and the rate at which it will change. Now, when we talk about populations and generations, it's important to consider that there are 2 different types as far as the structure or the time gap between generations. Some populations are semiparous, meaning they have non-overlapping generations. Others are heteroparous, meaning the generations overlap in a life cycle. So similarity, a lot of these are your R-selected species. In this case, individuals may reproduce only once in the entire lifespan, right? So here's one individual most of them will only have a lifespan for a year, some less than a year, but usually no greater than that. And in that course of that short lifetime, they have a single reproductive event. So there's only one moment in time where they're able to reproduce. And usually at that time, a lot of offspring are yielded. But again, it's only one event. So these generations don't overlap, right? So you have a parent population, that's generation one, we'll say, or the starting generation. They will last for a year, and after a year, they die. At the end of that year, you have a new generation. And at the end of that year, for that new generation of offspring, you'll have another new generation. They won't overlap. You won't have some individuals from one generation continuing into or remaining into the second generation. That's not what you see with, or that's more of what you see with heteroparity as far as overlap, right? So overlapping generations refers to heteroparity, where individuals can reproduce more than once during its lifetime. So there's more than one reproductive event or reproductive season during the course of its lifetime. And you see this with most organisms, particularly your K-selected, again, where they have these several reproductive events, and the generations produced from each reproductive event will overlap, right? So you have longer lifespans, much more than 1 year in duration. So individuals from that first generation may still be present and even reproductively active during kind of the reproductive era, per se, of that second generation. So you have overlap in generations, meaning some individuals from one generation are still around when you have individuals for that second generation. Now, based on these life cycles, that kind of determines how rates are estimated. Right? Rates will actually be estimated differently depending on the specific life cycle, whether this population follows life cycles that are non-overlapping or are overlapping. Right? So we're going to talk about both cases. Right? So estimating rates in non-overlapping generations. Right? So these are looking at species or estimating rates in species or populations of species that are similparious. Right? So our example is looking at an annual plant, commonly known as annual phlox or Phlox drummondi. This is a plant that's native to grasslands and open wood habitats in regions of central and eastern Texas. It's relatively small, usually grows between 6 to 9 inches in height. And it's particularly well present in southeastern states. It originally started as a cultivar species, has now since escaped garden beds and can kind of be found occurring naturally along roadsides and fields and waste areas. They're kind of notorious for their trumpet-shaped flower component. They're very fragrant, and they have they're 5 lobes, so 5 petals. And each flower has these little short, narrow tubes that appear in clusters at the stem ends, usually blooming from April to July. Now, The background story behind them is pretty interesting. The genus name is derived from the Greek word flux, meaning flame, in reference to the intense color that's exhibited, especially that brilliant red color that's exhibited by a lot of the different varieties of this flower type. The species name is in nod or honor to Thomas Drummond, who collected these plants originally in Texas and then sent the seeds from Texas to England. And he worked a lot with the reproductive function and genetics of this plant. We're working with it not because it's an interesting species, because it is, but more because it's a great example for a species that has non-overlapping generations. Right? So when we're estimating rates in this species, we're doing so by combining survivorship with seed production. Here, the offspring are represented by seeds. Seeds serve as the fruit of plants that will then go on to germinate and produce new offspring in favorable habitats. So this is a life table that's been created that also combines survivorship with the reproductive function or seed production of this plant species to generate or predict your net reproductive rate. So here, X represents age. For this plant, age is represented by number of days. So this interval is 0 to 299 days, 299 to 306, so on and so forth, for roughly equivalent to a year, a little short. This column represents the number surviving to day X, right? So thinking of, okay, the number that will survive maybe to the next day or to a week after. That's what this represents, or this number represents. Sorry. The next column, so this is our little LX, which should be familiar. LX represents the proportion of individuals surviving to that day from the original number. And then MX represents the average number of individuals kind of during that time interval. So this is representing the number of seeds per individual that are produced. And then this is these are the products of multiplying the proportions of individuals surviving to the average number of seeds that are produced during that time interval. So when you look at this table, first of all, you can understand or get a glean for life expectancy. Most of these plants will survive for only 1 year. And after 1 year, you don't have any more individuals left from that original starting generation. Right? So you start off with generation 1 that has, say, 1,000 individuals. By the end of 1 year, all 1,000 individuals will have died. That's what's meant by these non-overlapping generations. By the end of 1 year, moving into the next generation, there are no more survivors remaining of that original starting seed number or offspring number. Now, when we're using this information to generate the net reproductive rate, here the net reproductive rate is representing or exhibiting the average number of seeds produced by an individual in a population during its lifetime. Again, seeds are representative of the offspring of this plant species. So as stated before, you calculate your net reproductive rate by summing all of the LX values, which are the percentage of the population surviving at each age, times the average number of seeds produced by each individual for each age category. That's MX. And then you sum all of those products across each age interval. Right? So if we use that information, you generate the sum of these products, and that gives you an RO value, or your net reproductive rate, of 2.4177. So that means each individual leaves on average about 2.4177 offspring, right? So that's like an estimation of their true reproductive yield based on how many offspring they leave or generate that survive. Now, you can take this value and, using the kind of correlations that I described before, you can start predicting whether, or start describing whether this value indicates whether or not the population is growing or whether it's decreasing. So here, our value is greater than 1. And because of that, it indicates that this population is growing. So it will continue to grow in the future. Actually, I'll continue on with this. So because this is a non-overlapping population, we can calculate what's called geometric rate of increase. This is a value or a degree of measure that's calculating the ratio of the population size at 2 points in time, and using that as a determination of growth rate. Here, this factor is denoted by the symbol lambda. So you can only use this for non-overlapping populations because they will grow at a geometric rate of increase, meaning their RO value will be the same for every generation, right? So for N1, you will have an RO value of 10. That will be the same for every consecutive generation following. So you get more of this kind of linear increase or decrease in a population or population size. To calculate the geometric rate of increase, or lambda, you first have to determine or find this value, which is n representative of t plus 1. Nt plus 1 is the population size at a later time, right? So this represents the population size 15 years down the road. So 15 years or after 15 generations, what's the population size? That's Nt plus 1. You find this by multiplying the net reproductive rate, or RO, by a value called Nt. Nt is the population size at an earlier time, right? So maybe at generation 1, there's a population size of 100. You can take that value multiply it by the reproductive rate, right, which is going to be your rate of change in the population, to determine what a population size will be at some different time in um, in history, or in the a different generation. So you can figure out 4 generations, 5 generations later, what the population size is predicted to be based on that earlier measurement of time and the reproductive rate that's been calculated. So for and then once you find the nt+1 value, you plug that in to find lambda. So lambda, or your geometric rate of increase, is calculated as the division of nt, so that population size at an earlier time, divided by the population size at a later time. nt+1, when we're talking about our plants here, that would represent the total number of seeds produced by that population for that study year. So if we're looking at this age group here, right, we can take the number of individuals surviving to that day, so 996, multiply it by the net reproductive rate of 2.4177, and find out what the number of seeds that the population will produce and begin with the following year. Right? So if you do that math, you get a value of 2,408. Right? So 2,408 seeds should be produced and serve as kind of the starting number for the next year. In other words, your population size for the next generation, or the following generation, would be 2,408 individuals. Now you can go from there and calculate your geometric rate of increase, or lambda. Again, the ratio of the population size at 2 points in time. So for this example, you would do 2,408 divided by 996, which would give you 2.4177. So the geometric rate of increase for this population is a value of 2.4177. You can think of that as being almost like a slope value. Oftentimes, again, only in these non-overlapping populations, your RO value will be the same, or likely will be the same, as lambda, because, again, you have this kind of constant growth rate, or geometric rate of increase, for the population, as well as a continuous or constant reproductive rate. So these populations, again, they exhibit what's known as geometric growth, where growth occurs as these discrete increments, where each generation is essentially a discrete increment rather than continuous units. And you don't have individuals from this generation lasting into the next generation. This is the non-overlapping elements. So again, you see this with populations that have life histories where each generation reproduces a single time, or is simultareous, and then dies. Now, because of that, your reproductive rate, or RO value, which considers the birth of new individuals, it remains constant from one generation to the next. So because of that, we can use it to predict population sizes several generations into the future. It also means that you're going to have kind of this steady slope of either increase or decrease in population size or change, rather than something being exponential or logistic growth, which we'll talk about later in the video series on population growth. Now, how does this change for overlapping generations? How do we estimate rates in populations where the generations overlap with one another? Things get a bit different. So for this example, we'll use the common mud turtle. With your common mud turtle, it's been found that about half of the female turtles, it's actually 0.507 female, kind of that proportion of female turtles will actually nest each year. And of those females that do nest, most will nest only once during the year. Some will nest twice or maybe 3 times. But the mean number of nests per year for the nesting turtles is 1.2, which means that 20% of the turtles produce a second nest each year. When we talk about clutch sizes range from 3 to 4 eggs, with a mean clutch size. Clutch size is the number of eggs produced by a nesting female, being on average 3.17. So the mean number of eggs produced by a nesting female each year, you would calculate that by multiplying 3.17 eggs per nest times 1.2 nests per year, which yields about 3.8 eggs per year. So each female that does nest in general provides roughly 3.8 eggs every year. However, remember that only half of those total females in the population actually do nest each year, right? So of those viable females, only half contribute offspring. So the number of eggs per female per year is actually 1.927 eggs per female per year. On average, half of these eggs will develop into males, half into females. Depends on temperature of the nest and incubation temperature. But since in general the sex ratio of the turtle population is roughly 1:1, you can then take that 1.927 eggs per female, multiply it by 0.5 to calculate the number of female eggs per adult female in the population, which equals 0.96 female eggs. So the kind of proportion of female offspring that's contributed per female per year is equal to 0.96. Now, because the estimate rates, or because the generations overlap, and taking into consideration that half of the turtles nest each year, you can use similar information or matrices to calculate your net reproductive rate, right? So net reproductive rate, again, is going to be the sum of your LX times mx, or those product values across all age groups. And for our little turtle friends, that gives us a value of about 0.601. Now, that value, so kind of taking that value and using it to determine what's happening as far as the general trend in the population, since that value is less than 1, right? Again, this is our net reproductive rate. Since our net reproductive rate is less than 1, that means this population of turtles is declining. Because we're using these overlapping generations, we can go on to calculate something called generation time. Generation time is found by basically summing your LX, MX, and X values, right? So this is the product of your LX and MX, but also multiplying it for the year group, summing those values and using it or taking your net reproductive rate and dividing it by the sum of those products. That gives you a relative time value at which you can determine when new females can be contributed, or new female offspring can be contributed to that population. So more specifically, your generation time is the average time between 2 consecutive generations, between different lineages of a population. Now, in human populations, the generation time typically ranges from 22 to 33 years. But for turtles, using this formula, we find that the generation time is actually 10.6 years, right? So in general, there's approximately 10.6 years in between 2 consecutive generations. So you have generation 1, about 10 years later, you will have the establishment of the second generation of offspring from that first generation. So you can think of it again as a kind of almost like a population turnover as far as number of females in the population. Now, knowing your net reproductive rate and generation time allows us to calculate or estimate R. R being the per capita rate of increase, or your intrinsic rate of increase. Right? Again, we talked about this value before, in that you can interpret R as birth rate minus death rate. In most of these functions, it's more of a logarithm of net reproductive rate over generation time. But we sometimes call it the Malthusian parameter because it allows us, or reveals, again, the true rate at which that population is expected or will actually increase, depending on the certain parameters of that population. So it's more of an instantaneous rate of change in population size at the level of each individual. Right? So this is looking at the rate of increase per individual. And it's always expressed in numbers per unit time per individual. So it's always a measure of units over time or per unit time. Now, in a closed population, closed populations, you don't have the factors or contributions of factors of movement. So immigration isn't in play, emigration isn't in play. So in your closed populations, the intrinsic rate of increase is defined as the instantaneous per capita birth rate minus instantaneous per capita death rate, right? So simply B minus D, right? That's in your closed populations. In an open population, R is equal to births plus immigration minus deaths plus emigration. So you're taking into consideration the component or the influence of the movement factor. Now, when per capita births exceed per capita deaths. So if B is greater than D, the population is increasing. And this is noted by a positive R value, right? So if you get a positive R value, your population is increasing. If you get a negative R value, that means that deaths exceed births, or mortality rate is greater than natality, and your population is decreasing. If you had a value of an R value that was equal to 0, that would indicate a stable population. So if it's greater than 0, so it's a positive number, it's growing, equal to 0, it's stable, less than 0, or a negative number, it's decreasing. So if we calculated the per capita rate of increase for our little turtle friends here, right? Again, because it's non-overlapping, we're using the specific formula where you take the logarithm of the net reproductive rate over generation time. So for our turtle friends, we would do the logarithm of 0.601 divided by, or over, generation time, so over 10.6. And doing so yields an R value of negative 0.05. So because we have a negative value here, that means the population is declining. So birth rate is lower than death rate. You're not producing or having a high turnover of offspring that are replacing individuals that are lost, and therefore you're having a decrease in population.